once again, good morning everyone. So I would like to call Dr. Shankar Sharma from Uppsala University, Sweden. Nanowired delivery of cerebrolysin with mesenchymal stromal cells and monoclonal antibodies to neuronal nitric oxide synthase induced neuroprotection in Alzheimer's disease, neuropathology exacerbated by concussive head injury.
Thank you very much. First of all, I feel very honored to be part of this congress. I'm from Uppsala University, Sweden, and we are working on a joint project with Bureaus of Aerospace Research and Development in EU, together with US Air Force Research Laboratory regarding human factors affecting the biological changes in the body. And this research is supported by various organizations, and you can see Department of Defense, European Office of Aerospace Research and Development, and also we are working with Far Eastern University in Moscow, and also our colleagues are from Harvard University and various organizations. I am from Uppsala University. The main talk is we are willing to slow down the neurodegenerative diseases in general. That includes also Alzheimer and Parkinson's disease, and using nanomedicine, the latest approach to reduce the neuropathology of chronic diseases. These are our collaborators. Nasim Murasani from Romania, Rudy Kaslani from US, Alan Ojari from Harvard University, and Dr. Lefrente from Bilbao, Spain. Anka from Romania, she is now lecturer. Our colleague Ayan Tian from Fayetteville University in US, Dr. Huang from Beijing, Dr. Finn from Sichuan, Igor from Vladivostok, and also another Igor is also from Vladivostok. This is our student, and this is my wife. This is a huge collaboration. First of all, I would like to tell that whatever I am telling here, this is my personal views, and it does not represent the views of the organizations or US organizations and any other organization we are working with. Our main concern is our military personnel are vulnerable to Alzheimer's disease, and this could be exacerbated by traumatic brain injury and various other stressors. That's why we are developing the models and trying to study the effect of nanomedicine. The main component of our brain making disease is the blood-brain barrier that is protecting the brain from various insults. And in all other conditions, whatever happening in any kind of disease to the brain, they are leaky. It means that various components from the blood or external factors come into the brain and gives us neuropathological symptoms. Alzheimer's disease is one of the most prominent diseases of the mankind, affecting at least about 5 million people every year across the world. And so far, there are no cure or any medicine that can treat them well. As you can see here, the normal brain looks like this, and when the ventricles are expanded, as you can see here, then it can produce mild cognitive impairment, and its extent is increased, and that could lead to Alzheimer's disease. Another example, you can see the healthy brain is very compact, but mild cases, the ventricles are enlarged, and in severe Alzheimer disease, they are much more enlarged everywhere, taking out the functional aspect of the brain. Military is at very high risk of Alzheimer disease because they are engaged in war, and also they have chances more than civilians to get traumatic brain injury. As you can see that several papers are demonstrating that military risk factors for Alzheimer diseases. One of the main components is amyloid beta peptide that is not normally found in the human brain. People are healthy. But when, in various conditions, they are increased in the brain and slowly producing degenerative changes in the brain. This is an example showing the first one is the healthy condition and followed by other condition. You can see that how the color is expanding in various parts of the brain, taking all our vital functions and leading to disease. This is also an example, the healthy control brain. Here, traumatic brain injury and also other conditions you can see spread of disease by showing these pictures. The other example is that the brain is not affected entirely. Several regions in the brain affecting differently. And this is shown here, that this is the frontal cortex, this is the parietal cortex, this is occipital cortex, cingulate cortex, and temporal cortex. You can see the normal conditions appear quite similar, but post-traumatic stress disorder, like PTSD, is showing the maximum changes in the brain function. Traumatic brain injury also affect all these things, and when they are combined with traumatic brain injury and post-traumatic stress disorder, they are also showing much more changes, leading to severity of the cases. We are advocating that blood-brain barrier is one of the primary cause of Alzheimer disease, and we published papers in as early as in 1990s. And we are very happy to see that. To see this paper in PNAS that blood-borne factors are responsible for Alzheimer's disease. So this is certifying our theory. And this one paper from Netherlands shows a human cases that there is a leaky blood-brain barrier in early Alzheimer's disease. This is our experiment in transgenic mice model. We have shown albumin leakage in the brain of Alzheimer's disease, whereas when we have injected cerebrolysin, a commercial drug produced from Austria, that decreases the leakage of the brain. We measured biochemical amyloid beta, and you can see that this is amyloid beta in healthy cortex, and in Alzheimer's disease significantly increased. When we have given cerebrolysin, it is decreased significantly, and when we have used nanowired cerebrolysin, the increase was most significant. Another factor of Alzheimer's disease is the position of tau, and here is the normal, here is Alzheimer's disease, normal cerebrolysin, and nanowired cerebrolysin. So therefore, we try to play around this. Amyloid beta peptide 200 nanogram 30 microliter per day was injected into the animal cerebral ventricles for 4 weeks that produces Alzheimer-like symptoms. This is well known. Then we have introduced traumatic brain injury to see what happens. Can we enhance the pathology of Alzheimer's disease? Treatment, we have administered about 1 million mesenchymal stem cells. Now they are designated as mesenchymal stromal cells. So that's why this is old slide, it is still written stem cells. And then we have injected cerebrolysin and also the nanowired mesenchymal stromal cells. This is the model of head injury. So a weight of 114.6 grams is dropped over the right skull. That leads to massive impact injury model. As you can see that in this case, the left side is much more showing damage as compared to the right and blue side. In US military data, the head injury is much more serious, penetrating about 3,877 cases. Severe 2,469, moderate 40,449, mild 187,539, and not classified actually what kind of pain. So you can see the traumatic brain injury in military personnel are much more aggravated as compared to the normal civilians. Head injury alone can induce deficit in frontal and other areas of the brain, and the working memory is altered. This is right and this is left. We are telling since long that nanomedicine could be the key to resolving this approach to treat Alzheimer's disease. These are the nanowires. They are US patent by our friend Ryan Jian, University of Arkansas, Fayetteville, and the drugs are loaded on these nanowires like a scaffold, and they will administer. This is Dr. Ryan Jian, and this is Dr. Wang, who invented this nanowire technology for vision therapy. Research has shown that if even the stem cells can be nanowired, they can live longer, they can be better, and also their effect is prolonged. We show our observations that traumatic brain injury plus amyloid beta peptide induced Alzheimer's disease, the extravasation iodine was higher, and also the brain edema was enhanced. Brain injury, neuro injury, amyloid beta peptide deposit, and the leakage of albumin were also higher. When we deliver nanowired cerebrolysin, then it is significantly reduced in all parameters. We have nano-delivered mesenchymal stromal cells. The decrease was also significant. But when we have combined nano-delivery of mesenchymal stromal cells and cerebrolysin together, it is much more significant in reducing these parameters. This is an example showing cell injury. It is untreated. There are many cells that are injured. Nanowired mesenchymal stromal cells, some significant changes towards neuroprotection. Nanowired cerebrolysin also induced neuroprotection. And when we have combined nanowired cerebrolysin with nanowired mesenchymal stromal cells, neural memory is significantly protected. This is a model of mesenchymal that is normally altered in brain injury as well as in Alzheimer disease. This is an example how mesenchymal looks in Alzheimer disease and what is the status of large cerebrolysin protein, the astrocytes. This they would show the same thing, that blood-brain barrier disruption, inflammatory brain injury, and also after Alzheimer disease model, a nanowired cerebrolysin plus Alzheimer disease model, and the combination of all. The important thing is that the combination has reduced all parameters. This is an example of hippocampal damage. This is untreated, and this is cerebrolysin treated, and here is cerebrolysin and neprilysin treated completely. This neuroprotection was much more better here as compared to this image. Then another condition is stress that is occurring to military populations, and you can see therefore we have used resistant stress, that how it can affect Alzheimer disease induced neuropathology. This is an example of amyloid beta. You can see how much it is, and when we have treated with nanowired cerebrolysin, monoclonal antibodies to amyloid beta peptide, and another drug is significantly reduced here. In this case, also you can see that several changes were also reduced after the resistant stress and Alzheimer disease. The measurement of microglia is also significantly reduced in treated group as compared to the untreated group. Another form of microglia, you can see much more expression here and significantly reduced by the treatment in combination. At electron microscopy, you can see that there are much more wider spaces, damage of myelin and accumulation, and the treatment has not completely restored but also significantly restored as compared to the untreated group. So we can now conclude that significant reduction in neuropathology can occur by multiple combination of drugs and agents. And this is an example that they are real because there are 4 examples here. And some of the agents when nanowired are more superior as compared to the other drugs nanowired. So nanowired itself does not increase the potential of treatment, but you have to choose a particular drug or a specific drug to nanowire that could be more effective. And the possible mechanism could be that nanowire could penetrate the blood-brain barrier level, enter into the brain microenvironment, and can stay longer to protect. And this kind of research got an innovation award in 2018 by US, and the jury were several organizations from this. We are telling by different books our strategy to protect brain pathology from nanomedicine, and these are some examples published in 2019. This is published in Nature. These are again progressive brain research 2020, I guess. This is 21. And also we are talking about treatment of cancer using nanomedicine. These are International Review of Neurobiology. We are try Nanowired delivery. And so we believe that nano delivery of drugs and antibodies could be more effective as compared to the single drug alone. And we are working again to work on these lines. These are our sponsors and collaborators, various funding organizations. And I am from Oxford University. Thank you very much for your kind attention.